Brilliant. Thank you, Caroline, again. Slight change to the programme. So we're going to now hear from Professor Jeff Evans from Glasgow, from the Beatstone in the Glasgow, on some talks about his work that he's doing at the moment. And yeah, we'll hear from him. Thank you.
Thank you. I'd be very flattered to be called Mark Middleton, but Mark will be here later, I'm sure. Firstly, many thanks to the organisers for inviting me to come down from Glasgow. We've had a bit of a change in programme because I think Matthew Wheater is a virtual presentation and I've got to get back to Heathrow later on. So many thanks for asking me. And I'm just going to talk about some of the things. It's not going to be a comprehensive catalogue of all the trials that are ongoing. And it's good that we've got things to discuss, but I'm just going to give you a few selective highlights what I think is coming in the immediate future, if not here already. And because I'm a medical oncologist interested in drug development, I always start by disclosing the things I admit to in public, and these are some of the companies I've worked with over the last 30 odd years in developing novel agents. Now, this is a somewhat artificial separation of some of the treatment modalities we have in treating any cancer. We're familiar with surgery, and many of you in the room will have experienced that. Chemotherapy, radiotherapy, and then probably what I'm going to concentrate today is on molecular targeted therapy and immuno-oncology or immunotherapy. But of course, it's still somewhat artificial to separate them because clearly things like surgery and radiotherapy change the immune microenvironment, and clearly using things like targeted therapy and immunotherapy can potentially sensitize to other forms of conventional treatments as well. And it's not an either or, and it's quite likely we'll be using these, if not together, or in some selected combinations in the future as well. Now, this is the Hanahan and hallmarks of cancer and Weinberg. Now, this is the second iteration. I'm not going to talk all the way through this, but there's a number of molecular events that need to occur for a cancer cell to develop, and all of these do occur in a malignant cell. The molecule that drives each of these abnormalities will differ from one tumor to another. It may differ from one ocular melanoma to another ocular melanoma, and the real challenge is it might differ from one secondary deposit from another secondary deposit in the same person as well. So quite a complex thing. But I was struck by the slide earlier by the previous speaker showing what children think causes cancer. And actually most of those were correct. And most of them in some ways will influence some of these molecules. And the difference with the second iteration of this in about a 10-year period is the things we put on the outside. We now have drugs or methods of treatment that can target all of these molecular processes. The question is, can we identify what is the driver in any individual tumor and then target that tumor on that basis? And I think Tim Cook is probably going to talk about that later on this afternoon. So I'm not going to talk through the entire and try and explain this rather complex biology slide of ocular melanoma. Suffice to say that we know quite a lot about some of the key drivers, and hitherto it's been somewhat challenging to inhibit and block some of these abnormal signaling pathways. So unlike skin melanoma, cutaneous melanoma, we know that about 50% are driven by this thing called BRAF. And we have BRAF inhibitors, Vemurafenib and others as you will be familiar with. And of course the challenge there is that this is almost an anomaly to a certain extent, in that in skin melanoma, it's almost as if it's addicted to this particular pathway. In a lot of other tumor types, there might be aberrations, there might be abnormal genes, but the tumor may not be so addicted to that particular pathway. And that makes it more difficult then to try and inhibit and block some of those. So by example, we know that in uveal melanoma, over 80% have an abnormality in one of these two genes. But up to now, it's been quite challenging to try and block the downstream cascade that makes all the higher risk of cancer spreading and indicate for poor prognosis. And therefore we know that there's a number of players here. BAP1 is one of the very rare familial genes, for example, but these are present in over 80% of cases. And we're going to come back to that when we talk about some of the new drugs and the new signaling pathways. Now, so one of the key parts of that hallmarks of cancer is how cancer cells avoid being destroyed by the immune system. I mean, if the immune system was doing its job properly, it should get rid of the cancer because it's a foreign protein, and that's what the immune system does. But cancer cells are clever, and what they do is they have these things called checkpoints on the surface. And that is one of many ways in which the immune system can no longer recognize and eradicate the tumor cell. And if we look at the way we try and exploit the immune system to eradicate cancer, it's not quite as simple as we first thought, because firstly, you have to have what cancer-specific proteins, if there are any specific, to be released. They have to be presented to the immune system. The immune system has to be primed and activated. The immune cells then have to traffic to the tumor. They have to actually infiltrate and penetrate into the tumor. And there are some pancreatic cancer which is notorious for having this fibrotic coat that blocks those immune infiltration. And then you have to be recognized by the cancer cells. And the last step is really releasing the handbrake on the immune system, which is where these checkpoint inhibitors that you may have heard about, ipilimumab, nivolumab, pembrolizumab, etc., come in. So to try and correct all of those or try to exploit those is actually quite challenging. And I get quite cross with some of my colleagues when they refer to immunotherapy and immune checkpoint inhibitors like PD-1 and CTLA-4 interchangeably. There's vastly more ways we can exploit the immune system to eradicate cancers than just checkpoint inhibitors. They've transformed the way we treat certain tumor types like skin melanoma and others, but there's much more that we can do, and we're really beginning to scratch the surface of using the immune system to eradicate cancers. And people always ask, what's next? Well, I think we've got decades of work to try and better utilize what we've got already. The next thing is that interplay in that niche in how cancers spread between the immune system and what we call metabolism, cancer cell metabolism. How cancer cells actually use nutrients in order to spread, grow, and divide. And that's probably the next big thing that's coming after immunotherapy. Now, these are just some of the immune cell targets. We've mentioned a couple already, PD-1, nivolumab, pembrolizumab, CTLA-4, ipilimumab. You will be familiar with some of those drugs. But those are just very few of the immune cell targets, most of which we haven't really begun to try and act and target so far. So even if we think the striking difference that drugs like ipi, nivo, pembro, for example, have done in cancer, less so perhaps in uveal melanoma, but certainly in other tumor types, we've got an awful lot more opportunities, and this is just the immune checkpoint inhibitors, let alone the rest of the immune, how we harness the rest of the immune system. Now, at the moment, yes, we're mainly focused on these key molecules here, LAG3 beginning to come into skin melanomas. And these are antibodies that block and release that handbrake. There are others where you try and activate the immune cells and get these T cells activated and traffic into the tumor, which has a different form of toxicities, which we will have to learn how to manage as well. Now, it's not just about the tumor. I spent 30 years trying to eradicate the tumor, but actually we've discovered it's not all about the tumor. It doesn't go normal organ, normal cell, cancer cell. It's surrounded by abnormal stuff, which is called the tumor microenvironment, or the TME. And you can't go to any meetings these days without somebody talking about the TME, or tumor microenvironment. I'm slightly worried that we'll forget about the importance of the tumor as well. Both are equally important. It's not just about the tumor, it's the microenvironment as well. And there's a number of cells here that are involved in activating the immune system to try and get rid of the tumors, while there are those that actually are suppressing the immune system and restraining the immune system to stop it from eradicating the cancer cell. So there's this balance between immune cells in the microenvironment driving the cancer and those that are trying to hold it back. And how can we then manipulate those to sensitize the immune system and to those checkpoint inhibitors like ipi and nivo in tumors that are called immune cold and perhaps don't respond as well as things like skin melanoma, kidney cancer, bladder, etc. So I think we're just beginning to understand some of these cells, and these are a couple of cells here that are my favorite cells that we are trying to deplete within tumors that might sensitize to some of these immune checkpoint inhibitors in the future. Now, I'm just going to introduce this concept, and it's been mentioned already. This is a basis of what we call bispecific antibody, such as Tebentafusp. I still call it IMC GP100, so apologies if I revert back to call it what it used to be known as. And here you've got, it binds not just to a fragment of the T cells, but also binds to a part of the tumour. And just to show it in graphically here, so this is fairly unique T cell engager in some ways. You've got the cancer cell here, you've got the cancer molecule which has been exposed here on the surface. It binds to these various proteins here. And then we have the antibody which binds to these proteins as well as to the IMC-GP100, which is specific onto the cancer cell. And the very first study that we did, and I think it's about 2011, we started, and Mark Milton and I and many others were involved in the UK in the first in-human study. And we learned a lot about how to manage this drug. When we initially did it, we were dosing by body weight and going straight to the full dose. We now use that ramp-up dosing of weekly increases on day 1, day 8, and day 15 to abrogate some of the toxicities we experienced very early on in those initial studies. And to Immunocore's credit, they didn't restrict, as many trials do, they didn't restrict just to cutaneous skin melanoma. They also allowed mucosal melanoma, and interestingly, eye melanoma patients were also eligible. So ocular melanoma patients were eligible for that first study, which is relatively unusual because they're often excluded from melanoma studies, even in the very early stage. But they were allowed in, and we have an ocular surgical service in Glasgow, so we actually put in quite a few patients into that study who had uveal melanoma. And I was quite taken by the fact that we did seem to get some disease stabilization and responses, which was probably unexpected, quite honestly, at the time. And this is just to look at, at some of the early data that came out of the Tebentafusp study. And here we can see that if you look at the non-uveal melanoma patients that were in, this is, Mark was the first author on this paper, and we were involved in this study as well. You had the patients who had mainly skin and some mucosal melanomas, 65 of them, but the uveal melanoma patients did seem to have a better survival. Which was both intriguing and somewhat unexpected, and formed the basis of then a phase 2 study, and then a randomized phase 2 study, which is then Paul Nathan's paper from New England Journal of Medicine, showing here that we had an improved 1-year disease-free survival, overall survival, and progression-free survival. Now, it's worth noting when we come to discuss some of the other agents coming later on, that actually objective responses, now what we mean in cancer medicine when we talk about objective responses, is that you do usually a scan, usually a CT scan, before and then on treatment. And if you get a shrinkage of a tumor by a certain amount, say 30% of the sum of the diameters, that's called a response. If it's bigger by 20%, that's progression. Everything in the middle is stable disease. And as oncologists, we like to see responses, of course we do, and causing tumor shrinkage. But let's not underestimate the importance of disease control. Even if we don't shrink it by 30% on a CT scan to get that objective response, getting disease control that can last better than what was the comparator, which was investigator's choice, it was either Pembro, Nivo, Ipi, even dacarbazine. By getting better disease control, 1 year's survival is better, progression-free and overall survival was better, even if we weren't getting very high responses as we're accustomed to the tumor types. So this concept that getting disease control and how we best measure things on the scan, I think is becoming increasingly important, specifically in uveal melanoma as well. And patients were allowed to continue with the Tebentafusp beyond what would be conventionally considered progression on a CT scan if they were tolerating and clinically benefiting. And I think that's exactly the right approach to have taken to demonstrate the value of this study. So this, of course, is restricted to patients with a specific immune phenotype called HLA-A201, which I'm sure many of you are familiar with. And that's probably around 40%, 35 to 40% of the Western population. In some parts of the world, like Africa, for example, HLA-A201 is much, much less common. It's probably single-figure percentages. So this is for a selected population, a significant proportion of the Western population, up to about 40% who have that immune phenotype, which is necessary for drug action. So what is coming next? Well, we're back to targeted therapy here, and this is a somewhat busy slide. I mentioned earlier are those particular gene abnormalities, GNAQ and GNA11, and they drive downstream pathways. And there's something called a PKC, protein kinase C inhibitor, darovasertib, also known as LXS196 was the original name for it. And that blocks protein kinase C. But in common with many tumor types, there's a lot of crosstalk between those various signaling pathways. If you knock out one rung on the ladder, you can still bypass it. And therefore, you often need combinations. And we know that crizotinib was actually a drug developed to target something called ALK, which is about 4 or 5% of lung cancers. But it also blocks other molecules, as most of these targeted therapies do. And it blocks something called c-Met, which there's some good evidence to suggest that it might be an important player in preventing secondary spread from certain tumors, including uveal melanoma. And certainly the combination of the two gives a good tolerability. And in terms of objective responses, that's 45% in first-line treatment. In other words, patients who have not had any prior drug treatment for their melanoma, and about 30% in those who'd had prior and untreated patients. And in those with liver-only disease, about a third of patients had an objective response. Now, we have to be cautious with that, because if you have a phase 2 highly selected population, you can sometimes get higher responses than when you then go to a randomized trial. That's well recognized in oncology. And also, the comment that we made earlier, that perhaps sometimes disease control, even if you don't get the objective response, is important. Yes, if you target the immune system, that may occur. Here you're trying to hit various signaling pathways where you hope to see objective responses. And if you do, it then gives you the confidence to at least ask the question, is this superior to everything else in a randomized trial, before you assume that this is going to be the next standard of care. And we have been caught out in the past because many of you may be familiar with trials of taxanes in combination with MEK inhibitors like selumetinib. Many of us in the UK were involved in those studies, which were very promising in phase 2, but did not ultimately show their value in phase 3 trials for a lot of reasons that we've debated subsequently. But this is now certainly a very interesting combination in unselected patients, and which is currently being planned for global phase 3 trials in the first-line setting in inoperable advanced uveal melanoma. And those studies are about to start imminently. Now, we know that there are some targets both within the tumor and in the microenvironment, as we mentioned earlier. So how do they link together? Well, the answer is yes, they do. And there are various targets which are found within the tumor. PKC we mentioned, protein kinase C, which is darovasertib, c-Met, which we've mentioned, MEK, which we mentioned earlier, selumetinib, and these things called PI3 kinase inhibitors. Now, these are other signaling pathways, and again, these are complex with lots of crosstalk, so just knocking out one pathway, often you need combinations for the same reason. And there's probably 4 broad forms of these called alpha, beta, gamma, and delta. And delta is one of the ones that has been licensed for a form of lymphoma, but a lot of them are quite dirty drugs, and they cross many of these forms, beta, delta, gamma, so forth. And we know that certainly in the immune-associated targets, such as checkpoint inhibitors, we see this effect on what we call T-regulatory cells, which control some of the ways that the T cells try and infiltrate and eradicate tumors. So this is now looking at BAP1, which is one of the genes we mentioned in uveal melanoma. And if you have high BAP1 versus low, then clearly it has an impact on overall survival. And there's a number of genes which are significantly associated with loss of this BAP1, which is thought to be a poor prognostic factor by and large. And some of these will be representative of reasonable targets to be involved for potential research in uveal melanoma. So I'm going to skip over this one quite quickly in the interest of time, but we're working with a drug called IOA-244. It's just been given a name, and I confess I can't actually remember what it's been called, but I still call it IOA-244. This we presented in ASCO this year, some data from the trial, and I'll show you. So this is a highly selective inhibitor of PI3 kinase delta. And being highly selected, I think this is one of the reasons we seem to have had remarkably less toxicity than we would have anticipated from similar classes of drugs that may be not quite so selective against that particular isoform, against that particular subtype of PI3 kinase. So the study is called DION 001 is IOA244. It's a small company called iOnctura, based in Switzerland. There are only about a dozen and a half people in the entire company. A lot of them used to work for CRK various laboratories. And we've one of two centres, along with a hospital in Italy, in Siena, that have done these early studies in uveal and other tumour types. So this was the study. In the first part, we did the classical step of finding the dose and schedule, starting off at a low dose and increasing doses up to 80 milligrams every day, given on daily administration. We didn't do what most drug development trials do, which is dose up to toxicity. The conventional way of dosing, certainly for chemotherapy drugs, is to determine dose-limiting toxicity. You increase the dose in subsequent groups of patients until you found the right dose based on toxicity. Now, that works okay for chemotherapy drugs where there's a link between dose and toxicity, and possibly between dose and effect on the cancer. Whether you need to do that with immune-targeting agents or targeted therapies is a very good question, to which I don't think we know the answer as yet. But certainly the FDA now have introduced something called Project Optimus, which says, do you actually need to give big doses to toxicity? Can you get away with less dose, less costly, less toxic, and maybe better tolerated in the long term? So a lot of drugs now, we're going back and re-evaluating lower doses to see if they're more appropriate. Not relevant for chemotherapy, but certainly for this sort of drug. So we've actually landed on a dose of 80 milligrams without seeing any dose-limiting toxicity. But we did see an effect on, at least on the biology of the cancer. Patients were having regular blood samples, but also having a biopsy of the tumour before going on to the study and on study. So it's mandatory pre- and on-treatment biopsies of the tumour to really look in depth at the tumour to see what effect we're having biologically, to demonstrate that we're at least seeing a biological effect, even if we're not seeing an effect on toxicity. And having established that, there was an initial plan to look at this alone and in combination in multiple tumour types, but actually we've just landed on completing only a uveal melanoma study, and we're just wrapping up the study and already planning a pivotal, possibly registration study in the beyond first line, in the second or third line. So already we're beginning to talk about first, second, and even third line treatments in uveal melanoma with drugs in inoperable disease, which was unheard of probably even 5 years ago, when we were really looking to see better drugs than we had when first we had Tebentafusp. Now we're probably going to have the daraversatinib, crizotinib combo, and probably IOA and others coming up behind it. So this is just to show toxicities at the various doses, and grade 1 we called mild, and as you can see, we see very few mild toxicities. We see moderate, but any severe treatment-related side effects, we're not really seeing anything. So we are seeing some toxicities, whether it's related to the tumour, related to the drugs, always difficult to know for sure, but none of these would cause any concern regarding dose limiting. Yes, they're important for patients. We're not undermining or underplaying the toxicities experienced by individuals, but in terms of identifying a safe dose and schedule and a drug that seems to be remarkably tolerable, then certainly I think 80 milligrams a day has certainly been that. And just to give you an example, I have one patient who I think next month will be coming to see me every 4 weeks for it'll be the third year on his trial in October. So that just shows you the tolerability, as well as I think we're doing something for the natural history of the disease. Again, that protracted disease control, even if we're not getting objective responses, a bit like we observed with Tebentafusp. So this is just showing some effect on why we landed on 80 milligrams. We are seeing effect on these T regulatory cells I mentioned earlier, which is one of those key parts of controlling the immune system and those other molecular targets. And both in the tumour and in the blood, we're seeing a decrease in these T regulatory cells in those who respond versus those who don't respond. So clearly we're having a biological effect. What are we doing in terms of effect for patients' tumour? So this is what we oncologists like to call as a swimmer's plot. So you can imagine looking down at the top of a swimming pool from above, and in each of the lanes represents each patient and how long they've been on treatment, and with an arrow, are they ongoing? And this is these are some of the patients who are on, and their previous responses and what they were on before in grey. And you can see patients on short-term exposure to IPI, Pembro, IPI, and IPI and Nivo here, or no treatment, and certainly getting durable disease control, even if not meeting that objective response criteria on scan. And what everybody says, well, what does that mean? Good question. But if we look at what happened beforehand and their previous disease control duration on conventionally available treatments like Ipi, Pembro, Nivo, certainly this is an encouraging disease control that we've seen so far in a limited small number of patients and highly selected. So the usual caveats around that, but I think it raises questions that we really need to interrogate in a randomized trial. So this just shows some of the survival data, and as you can see there in part A, this is dose escalation. As we ramped up, those are the survival data. We get in 2/3 of the patients at 6 months. We haven't in part B, where we've just looked at uveal melanoma patients alone at 80 milligrams. We haven't got long enough duration for out to 1 year yet, but we are seeing a very encouraging 4 and 6 month survival as well as overall survival. Again, non-randomized, so the usual caveats about that, but I think what it does is a go-stop-go decision about should we take this drug forward? Yes, I think we should. And should we compare it against other standard of care therapies beyond first line, beyond Tebentafusp if they're HLA positive, beyond crizotinib, Davo in patients who go on to that study. So where are we going in the future? Well, everything in oncology, we always, when we're developing new treatments, start initially with advanced disease, i.e. disease that cannot be operated, patients who fail standard therapies or for whom there is no standard therapies. That is the conventional way, rightly or wrongly, of looking at new drugs. But so there are a number of new drugs. I mentioned darovasertib and crizotinib, IOA 244, which I'm working on, so we focused on that today. Optimal combinations. Some of these may be combined, for example, with Tebentafusp. I know PRAME is another immunocore targeted agent, again in the same HLA group of patients, which is being explored alone and in combination with Tebentafusp in multiple tumor types, including uveal melanoma. One of my colleagues is leading on that for us in Glasgow. And as well as all of these for advanced disease, we shouldn't forget about how we can target liver-only disease with local regional, and I know Matthew and others are probably going to talk about chemosaturation and DELCATH, which I gather has been mentioned several times during the day, understandably and rightly so. So are there ways, microwave ablation, other ways of treating liver-only disease without necessarily resorting to drug treatment? So I think we should be glad that we've now got at least several potential options here that we can begin to work with over the next few years and next decades or so. And there are many others being developed right now. Of course, that's an advanced disease. The other question is, can we take people who've had their primary tumor radically treated, potentially curative treatment, but at medium or high risk of recurrence, and try and reduce the risk of recurrence, delay recurrence, and improve overall survival that way, i.e. preventing recurrence, adjuvant treatment as we call it, well-trodden path in breast, lung, bowel, stomach cancer, etc., etc. Not so in uveal melanoma, but there are already studies, Tebentafusp and others, beginning to explore in the adjuvant setting after either surgery or ruthenium plaque to the primary to try and reduce the risk of secondary spread. And I suspect Mark is going to tell you about how we might monitor that with circulating tumor DNA to try and identify patients before they have any visible evidence of recurrence radiologically, for example. And how we might use that as monitoring. And I've already mentioned earlier how we have the challenges of identifying whether drugs are effective in advanced disease when you get this durable disease control, which is great news, without necessarily that shrinkage. And there's an entire new way of looking at imaging now called radiomics, which you need an expert radiologist or physicist to be able to help you with these studies. And this is something we do in the IOA 244 study. And in fact, there's an abstract popped into my email as I was on the airport on the way down this morning of some data we want to present in another meeting later on this year, where we've looked at different features rather than just the shrinkage of the tumor, but the nature of the tumor that you can see on a scan to see if that gives you better clues to what's actually happening and how we might identify the right patients to get the right treatment. So we're looking at after a radical treatment and even before radical treatment, can we give drug treatment prior to perhaps enucleation, so to spare the primary organ and make primary treatment less morbid without losing an effect on overall survival, for example. So I think there's a number of approaches now, and like the rest of oncology and other tumor types, we're playing catch-up with new drugs in new clinical scenarios first, second, even third line, as well as pre and post-operative. So I think there's a great opportunity now. And the reason we're doing all this, yes, I've got excited about the biology maybe this afternoon, but the most important thing why we do this is to try and improve quality of life and overall survival for our patients. And this is a team effort. We need the goodwill of patients and their carers, my colleagues in the Discovery Science Labs, those in the clinical trial infrastructure, and charities which support patients who are going through this disease and going through clinical trials. And if I can make one last plea for clinical trials, it can have unexpected benefits. I enjoyed working with my clinical trial coordinator so much that I ended up marrying her, and now even the family pet is involved in Stand Up to Cancer, and has got a lot of insight into my shortcomings and how we manage cancer. With that, I'll happily take any questions if I've not gone over time. Thank you.